Hi guys, welcome back to another episode of Tambi's Guide to Not Effing Up A-Level Biology. Today we're going to be doing actin, myosin and the sarcolemma. This topic's a bit iffy, but once you get the hang of the mechanics, it's actually not too bad. So this is going to be, well, I'm hoping it's going to be a relatively quick video, but let's crack on. So first of all, we're going to start talking about the muscle filaments themselves. So in a muscle band, I'm sure you're pretty familiar with this diagram, but you have a bunch of thick filaments and you also have a bunch of thin filaments with a few areas of overlap between the thick and the thin filaments. So we're going to start talking about the thin filaments first. So we do a little arrow, thin filaments. And the thin filaments are made of actin. I'm sure you know that through your revision, but I'm just going to go over the structure anyway. So actin is kind of these two chains made up of amino acids, so effectively polypeptide chains. Actin is quite a big molecule, but this is quite a simplification, but that's all you need to know. So this is a chain of, let's say, actin. Remember, it's a really long polypeptide chain, and you have two of them, so I'm just going to put a 2 there. And the way I like to think of it is a bit like collagen. Obviously, it's not collagen because it's not 3 polypeptide chains, but it's forming that structure that's intertwined, and you can see that precisely over here. Another thing that you need to know is that actin is laced with tropomyosin. We're going to learn more about tropomyosin in the next video, but for now, all you need to know is that actin is part of the thin filaments and it's laced with tropomyosin. So, laced with tropomyosin. You probably already know what tropomyosin is. If you don't know, you're going to see it anyway on the next video. But yeah, laced with tropomyosin. So that's the thin filaments sorted. We're going to talk about the thick filaments now. And as you've guessed, the thick filaments are made of myosin. And myosin has quite a similar structure to actin. The only difference is you have a bulbous head here. So we're going to see the significance of the bulbous head again on the next video. But yeah, it's like 2 polypeptide chains kind of meshed together, and it's almost wound together. I think that's the terminology you use, so wound together. Again, it's 2 myosins that are wound together, and we have this bulbous head. We're going to learn, again, the next video will interlink with this, but the bulbous head is for ATP detachment and making sure you can have that those filaments moving across each other. And I think that's all you need to know, actually. So I'm just going to do a quick recap. This is actin, 2 polypeptide chains wounded together, laced with tropomyosin. Myosin, 2 polypeptide chains of myosin wound together. They have the bulbous head, which is characteristic of your muscle contraction. This is just called a tail. It's not really any significant word for it, just call it tail. Anyway, so just to start off, the structure of actin and myosin. Now we've got that sorted, we can start talking about the really dodgy terminology that comes through. I don't know why biologists decided to name it like this. They just like using terminology from other languages, but we can't really do much about it. So I'm just going to redraw that diagram over here. Thick bands, and I'm going to draw out the thin bands as well. Do them in red this time. And let's start labeling it. So let's start with the obvious ones. The line in the middle is called the M line. It's self-explanatory, it's in the middle. I'd also like to mention that this whole thing, we call this a sarcomere. when you have a muscle fiber and you have a myofibril, it's actually made up of a lot of sarcomeres. It's a bit like a polymer, keeps going on and on. So this is one sarcomere that we're going to start talking about. So the M line, it's in the middle of the sarcomere. We have these areas, and they're called Z lines. So this is where it gets really cheesy. The way I remember Z lines is they're on the opposite sides. So they're on the opposite sides of the actin and myosin filaments. And as a result, you can see that they're on the terminal ends of the sarcomere. So the opposite sides. Cringe all you want. This is how you try and get any stuff. I don't know, I'm trying. No guarantees, but yeah, the opposite sides. That's the way I remember it. Okay, let's now start talking about these areas themselves. So the area over here that consists just of the myosin or thick filaments, we call that the H zone. So I might do it on a different color actually. Blue, let's do this one. So this area here, maybe I can do like a little dotted zone around here. This is your H zone. Only contains myosin filaments. I mean, there's not really a way. I just remember that the H zone's in the middle. I use the other like terminology stuff to like kind of remember it. And that leads us to the next part, which is going to be the A band. Now, the A band is all of this from here to here. And you can see it's between the regions of overlap. And so the regions of overlap here and here between the actin and myosin filaments and the whole H band, H zone itself. So, sorry, this is a really bad diagram. But yeah, let me just do a little arrow here. So this is called your A band. And the way I remember this is A for all. So it contains all of the actin myosin filaments and all of the H zone. That's the way I remember it. So that's the A band. And this part here, or these two actually, these are your I bands. And again, another way I like to remember this is I for individual. The I bands only contain actin filaments, and they don't contain any myosin filaments, so they're kind of individual in that sense. So I bands because they're individual. So I'm just gonna like sum this all up. So I'll do it in blacks just so you can remember it. So let's do the M line, middle of the sarcomere. Self-explanatory. You have the Z lines, Z opposite sides. Hence they're on the terminal ends of the sarcomere. I'll just put that there. Terminal ends of sarcomere. There we go. A band, A for all, in the sense that it contains the overlap region between the actin and myosin, and also the H zone, which is only the thick filament / myosin. We have the I bands, or the individual bands. It doesn't actually stand for individual. I just remember it this way. Individual because it only contains actin filaments. And that's the diagram there. I mean, as I said earlier, it's actually not too bad once you get a hang of the mechanics, but Yeah, these are like the ways I try and remember it. When I remember when I saw it first, I was reading off a textbook and it was so foreign that like you just can't remember it that well. So this is just how I've kind of adapted to like learn it. The H zone, this like I haven't really thought of something like the others to like kind of remember the H zone. But as long as you remember the other stuff, you could almost eliminate it down to the H zone, which has to be the region containing only myosin, which is this region over here. And that's it. I think this is like a shorter lesson. The next lesson is going to be pretty long because it's going to be about the actual process of muscle contraction. But there you go. Actin, myosin, and the sarcomere. Hope that helped. Thank you.